to understand what these medications are doing, I guess we should probably start with like Digestion 101. I eat food, and then what happens?
Yeah, the relevant part to digestion, I suppose, would be to look at, let's assume you are responding or you responded to some sort of hunger cue.
Yep.
Your brain, therefore, is very much involved in, kickstarting this process and interacting with the surrounding environment. You eat that meal, whatever it is, depending on what the meal was made up of. You eventually break that meal up into its remnant parts mechanically and enzymatically.
Mechanically being chewing, enzymatically being just digestion, the enzymes in your stomach.
Mm-hmm. So mechanically, yes, can I suppose be considered chewing, but also when you think about how your esophagus actually pushes the food down, you're not consciously thinking of the food going down, but there are these contraction-like movements that continue to move the food.
Makes sense. Okay.
Down further. So yes, there's that component. Eventually the food is broken into its constituent parts: glucose, amino acids, fatty acids, amongst other products as well. Now, let's pretend that the food and these parts have made it to the small intestines. That's where I think the crux of this conversation lies, is just given the hormones and the signals and such that's in the absorption that occurs at this stage.
So it mostly happens in the small intestine?
So, right. So most of your nutrient absorption occurs there.
Okay.
Yes. And so if we also pretend we're under normal conditions, so we have normal blood glucose, we're not pre-diabetic, diabetic, no other conditions that we know of. In general, the glucose absorption into the bloodstream at this point through the walls, basically, of the small intestine triggers insulin release from the pancreas.
Okay.
So we're going to talk about some key hormones here: insulin, glucagon, amylin, GLP-1, of course, GIP. And I not that you need to write those down, but just more or less to just be ready for those, as these are 5 of probably 500 key peptides, key hormones in this whole spiel.
Okay. All right. So we eat the food, we start to digest it. It gets broken down into base components, the things that our bodies can actually use. That gets absorbed through the small intestine wall, largely. And then that triggers the release of insulin.
Mm-hmm.
Okay.
Mm-hmm.
What is the insulin doing? What is it doing functionally for our body?
So in this case, and in general, it's a key hormone that's involved in the regulation of glucose, cell growth, and metabolism. So it's sort of a, when we start to really talk about how these drugs are working, in some sense, insulin is very much involved, mechanistically with these medications. So essentially, to take it one step further, insulin functions as a storage hormone. So the way you can think of this is that it actually promotes the uptake of the circulating glucose that's now in the blood, out of the blood and into the cells, where it can eventually be stored away in the liver and skeletal muscle, adipose tissue, etc. And this is, again, under normal conditions, we want to start to take some of this glucose from our meal out of the blood, so we don't find ourselves in a pathophysiologic state, basically.
Okay, so the insulin is signaling to the body, hey, there's glucose for you. Mm-hmm. Take it out, use it.
Yeah,
got it.
Yeah, and even I know we'll kind of get to this point, but I'm sure people are probably thinking this sounds more like a discussion on potentially diabetes,
right? Yeah,
but the whole premise there is that the GLP-1 receptor agonists, again, as an aside, were originally developed more or less to treat type 2 diabetics because of this role, how it affects how this peptide, ultimately affects insulin release, amongst other things. But, bear with us as we
got to
chip
unpack
away
a lot.
at some of lay the groundwork.
Okay. So you said insulin, and you've talked about glucose. What are the others that you mentioned, the other hormones that are going to be relevant to the conversation?
So in addition to insulin being released from the pancreas, there's also something called glucagon that is simultaneously decreased in this circumstance that we're talking about. So
they counterbalance each other.
Yes, more or less.
Okay.
Yeah. So they have generally opposite actions. This particular hormone has a lot of different functionality in the body, but again, in this context, again, just serves to do the opposite of insulin, which is to stimulate glucose. So in general, it's stimulating the production of glucose from the liver. But in this case, since it's being decreased, there's less glucose being produced for a reason.
Okay, so the liver can also produce glucose, I assume from stored glucose or stored fat. And that's like when you're hungry, but you don't have food, that's when your body ups the glucagon to give yourself the energy that it needs to keep functioning.
Mm-hmm. Yeah, there's a lot of there are a lot of situations in which your liver, your body wants to ramp up the mobilization of glucose. If you haven't eaten for a while,
right,
or if you're exercising and you're starting to deplete, your energy stores, so to speak. This is all of these sorts of metabolic processes are very much tied together, and they're all kind of simultaneously happening and changing and feeding, right, feeding off each other. So
it's not just on off things, right, happen and counterbalance each other constantly. And mm-hmm. Okay. What were the others? Amylin, you said?
Yeah. And so before we get to amylin, okay, the last thing I forgot to mention about glucagon is just that this decrease in glucagon and increase in insulin just basically allows insulin to better exert this blood glucose lowering effect, because in this context, that's ultimately the purpose of what's needs to happen. And there's a multitude of, I know we narrowed it down to 2 hormones there, but there's just a lot.
Right.
There's a lot more than just those 2. But amylin is another key hormone that's co-released with insulin.
Okay.
And this peptide, you'll see why it makes sense. This peptide basically serves an important role in meal-ending satiation. So it signals to you as you're eating or as you finish a meal that, all right, I'm full, I'm good, I'm satisfied. And so as these types of hormones are more or less dysregulated in the setting of diabetes or obesity, and that you see why, again, because these drugs act on these peptides in some sense, why it can make a difference.
Okay.
But in normal conditions, amylin helps to slow gastric emptying, which in and of itself has a downline effect on mealtime blood sugar, mm-hmm, and also fullness. And so by slowing gastric emptying, we, that's just, all that really is just food going from the stomach into the small intestine. And, that has, you can essentially feel that physically, in addition to the underlying physiologic stuff that's happening.
Got it. Okay. All right. What was I feel like there was one more.
So the big one, save the most important for last. Well, 2, we have GLP-1. So glucagon-like peptide 1, it is one of 2 incretin hormones. The other is GIP. And so the incretin effect, just as a bit of background, that was actually discovered in research years and years ago when they noticed that there were differences in insulin secretion between oral glucose intake versus parenteral intake or intravenous intake.
Oh, interesting.
And so it's kind of just sheds light on, as you consume foods orally, there's just a lot more happening, and there's a lot more, hormonal interplay that's occurring, as opposed to just this, well, I eat it and I should be full because I had enough nutrients and the food volume was high enough, so therefore I'm satisfied. It's just that there's just a lot more, there's a lot more there.
Okay. So you said versus intravenous. So like literally when somebody's not able to eat for whatever, mm-hmm, reason, if they give them intravenous food,
yes,
that it doesn't release all these hormones in the proper ways,
just has
in the normal ways. Mm-hmm. And so, okay. Cool. Interesting.
Yeah. There's probably a better way to describe that effect, but more or less that's just the category of these 2.
Okay. GLP-1 and GIP.
Exactly.
Okay.
Exactly.
I will not call them glucagon-like peptides or whatever it is that they're actually called. We're going with the initials.
You can do it.
No. My track record with drug names alone is sufficiently bad that we're not going to confuse people.
Add it to the real.
Yeah. Anyway, okay. So we got GLP-1 and GIP. That feeling of fullness and satiation, you said that comes from amylin.
Yes.
Primarily. and from the physical sensation of the amylin release helps you to feel full, and it slows down gastric emptying, meaning food leaving your stomach going toward the small intestine. It slows that process down. Therefore, you just feel fuller. Mm-hmm. Got it. Interesting.
Yeah. And so to piggyback on those effects, GLP-1 in essence has a similar effect. This is another example of a peptide hormone that's, has the potential to also further delay gastric emptying. And so this is also, again, another reason, don't want to just gloss over this. This can help, provide a more steady, a more steady glucose absorption to occur, as opposed to quick, sometimes it's the transit time that has an effect of how quickly or not so quickly we're absorbing, nutrients into the blood.
Okay.
And GLP-1 also lowers glucagon secretion. And if we remember from just a little bit ago, in doing so, less glucose is going to be mobilized into the blood.
From the liver, from the stored. Okay, okay.
And the key, I think the key part here is that there are GLP-1 receptors. So the GLP-1 needs the GLP-1 receptors. Insulin needs insulin receptors.
Sure.
The key is that these receptors are all over the body. They're in the small intestine, the large intestine. There's some in the brain. And so this is why, when you talk about, diabetes, and you talk about obesity, and you talk about these, we talk about hunger signals and interacting with your environment and all these different things at play, why, weight loss and food consumption has more, has a wide reach.
Yeah.
And so the brain is very much involved.
Okay. All right. What are we missing? Anything else in this conversation?
Just a couple points, just because related to GLP-1, that's still, in terms of when we're relating it to the medications, the main hormone here that we're talking about, just a couple things biologically. It has a half-life of 2 minutes. And we'll get into these things more when we get into the drugs, but a half-life of 2 minutes, your body, there's not much time for there to be activity for it. So when you look at the medications, what they're essentially end up doing is prolonging that activity. The other thing that I found really interesting is that in type 2 diabetics, GLP-1 activity is not so much affected, but GIP is affected pretty dramatically. They did different studies in knockout mice, essentially means they removed the genes for it, and they could see that without it was linked to increased obesity and whenever they didn't have those genes. Also in the brain, GLP-1 is involved in neurogenesis, so essentially creating new neurons in the brain. It has neuroprotective effects. Also reduces cell death. So there's a lot, they're finding a lot more, even related to potential targets for GLP-1 and Parkinson's, Alzheimer's, stroke. And I think whenever you're looking at type 2 diabetics, we've got the whole cascade of health issues. So now you're throwing in that this, GLP activity in the body, exogenously from a medication can have effect on other chronic long-term conditions that often with aging come into play as well. So again, these are not well studied at this point, but I think it was pretty amazing to see it dovetail into these other chronic conditions as well as the neuroprotective effects. GLP, whenever you look at its activity, also is protective to the beta cells. So it helps to proliferate the beta cells. Beta cells are what release insulin. So when you look at like the entire feedback loop, it's pretty amazing, again, that it's involved in the production in that way and the health of the cells. The other thing that Dante hit on it, but in the study that I was reading, they use the word, it decreases inappropriate glucagon secretion. Because if your body still wants to maintain homeostasis of blood sugar, that's what it's trying to do. So if it completely inhibited, so now you've got this 2 minute hormone that's naturally acting in the body, now acting long. If it just was totally a blocker of glucagon secretion, you could get hypoglycemia. Yeah. So it has a regulatory effect. So the more we learn about it, the more you get into it, and you start getting into things that are regulatory, it really is amazing.
That I think is one of the big things is the more I learn about all this stuff, I'm realizing just how incredibly complex these systems are and the way that everything seems to affect everything else.
Yes.